This project is really about, I think the major goal is making an antibody test. That you can use the blood to test the antibody, whether the individual have contacted with the virus and recovered and has antibody generated, so they would have some type of protective immunity. So there are a number of tests out now. Not a lot of them get approved already by either the FDA or by Health Canada. But majority of the tests are reliant on a so-called single antigen to capture the antibody. So we know the virus contains multiple proteins, and the antibody can be, in principle, against any of the viral proteins. So that you're going to miss some with a single antigen. So what we are doing is to use a combination of different antigen, but we're not actually using the whole antigen protein. Is using the pieces of protein so we can put a lot together. So in that case, we have more of an inclusive, kind of mixture of antigens that can capture all the antibodies, all types of antibodies that are produced by different individuals. The antibody test usually is not fully initial diagnosis because the antibody takes a little bit of time to develop, usually starting about day 7 or so. Then it gets stronger in week 2 and even stronger afterwards, but kind of wears down in a few months. So the window of antibody testing is pretty large compared to the so-called RNA testing. So I think that the window opportunity to apply the antibody test is pretty long, and we can use that really track down people who might be actually asymptomatic. It's a great thing that you see so many scientists in the world are doing similar things. I think we are kind of compete against the time and compete against each other as well. I know that there are people who are doing similar approaches to us, and it's not identical, but we are doing similar things. And the common goal is really to develop something that is very, very powerful and with high specificity, high sensitivity. So that's our goal. And then the timeline, we would like to get it tested as soon as possible. And then we are just making our first prototype of the test. So we're gonna, running our trial test next week. Oh, this helped us tremendously because I think that all the reagents are quite expensive, and then some of the control substances we have to buy from commercial sources to speed up because we cannot do everything in our lab. So we want to do the most important things in our lab, and then and use commercially available resources to speed up the process. So the funding is really important, and it also allow me to hire people specifically to work on the project. I want people to really stay, optimistic. I think everybody's working very hard, including us. I think, of course, the frontline doctors and nurses and those people are really putting their lives on the line to help others as well. So we are doing our part, and then we think that we're going to find the good test, and then to evaluate the people, and then really to identify people who may have a protective immunity. And I know other laboratories are working on developing vaccines, and our research also can inform that kind of effort as well. So I think stay hopeful is the message I want that people know.